Hello, am I audible? Mm, can one of you tell me if I'm audible? Hello, hello. Please let me know if I'm audible or not. Hello, Miss
Yes,
Anisha
ma'am.
Kole, am I audible?
Yes, ma'am.
Okay, so we'll wait for like 2 more minutes and then we can start. Yeah, I actually wanted to ask you something. Um, were you guys receiving the mails for the Monday's live session? Because the last two times I didn't receive it. So let me know if you received it or not, because I had raised a complaint about the messages. Even though this is the last class, today I got the mail regarding this. Let me know in the chat box, or you can talk in your mic and tell me if for the last 2 3 sessions if you received the mail for Monday live session. Anyone? Okay. I think we can start now. Let me just share my presentation. Yeah. Is my presentation visible? Someone please let me know on the mic. I won't be seeing the chat box when I'm in presentation mode. Can someone tell me yes or no? Are my slides visible?
Yes, ma'am.
Okay, good. So we'll start with week 8 live session. This is Cell Culture Technologies from NPTEL. The course instructor for this is Professor Manak Das from IIT Kanpur, and I am Sumati Raviraj from University of Hyderabad. So this week the module was Advanced Cell Culture Modules 1 to 5. So this is what you would have watched the videos. So let's just go into the questions because this time the theory questions are a lot, so we can discuss few things within the questions itself. So this is from your assignment. The but I want you all to give me your answers for this. So it should be an interactive session, and also because maybe if you would have attended the first two live sessions, if something was similar to this, so let's just interact. You guys first tell me the answers if you know, or you can guess the answers, and then we'll go to what is the right answer. Okay. So the first question is, during depolarization, membrane potential shifts from A negative to positive, B positive to negative, C remains unchanged, and D none of the above. What is the answer for this? Can someone answer in your mic? Yes, tell me. One of you can at least guess the answer if you don't know. Anisha, Pranab, Soumya, Deep. Okay, you guys are typing in the chat box. I told you when I'm in presentation mode, I cannot look at the chat box again and again. You saw I have to close everything and go back. So please turn on your mic and give me the answer. So someone said A, someone said B. Can you please tell it in your mic? Yeah, if you don't answer, I we will not be going ahead. At least give me some guesswork here.
I think it's negative to positive.
Negative to positive. Okay. You took so long to answer on the mic. You have to please answer on the mic because I can't go back for every question in the chat box. Kindly turn on the mic and answer. Very good. Your answer A was correct. So, what is depolarization? Depolarization is a change in the distribution of electrical charge within a cell, the difference in your charge within the cell from outside to inside. So, that time it is essential for the cell to maintain this membrane potential. So, the change in membrane potential is very important for certain stimulus or impulses to act, especially in your action potentials. So that time your body will start to respond according to your membrane potential. So your resting membrane potential, what should it be? It should be almost around minus 70. Okay. So that is your resting membrane potential. So it's not like all the time your membrane potential will be exactly minus 70. So it is dynamic, right? Because it is maintained because of the ions which are outside and inside of the membrane of the cell. So this minus 70 is not exactly minus 70 all the time. It might be plus or minus variations, and it keeps changing according to the flow of ions. But there is a buffer kind of range in which it is still known to be in the resting position. So this threshold will be broken if it changes more than minus 55. So your minus 55, here you can see in this graph, it is marked in your purple dotted line. So this minus 55 is the threshold. Once this threshold is changed, that means if it is going above minus 55 because it's negative, if it's going to some minus 40, minus 45, something like that, then it will cause a depolarization. That is called depolarization. So this will be a immediate stimulus, and then this depolarization will carry out the ions, or it will move the signal or wherever necessary. Okay, so this is your resting membrane potential. Then there is a threshold. This is your threshold level, and from there, if it increases due to some stimulus, there will be your depolarization, this increase. So it is going from minus 70 to plus 30. So it's going from negative to positive. Okay, so what is the definition? Like we read here, the difference in the electric potential between the outside and inside of a biological cell. This is a simplest definition. The units, it is measured in millivolts, and it's typically, it will range from, this table is for your membrane potential. Okay, it's not for depolarization. So you should remember, this is only for membrane potential. This was your depolarization, this was your depolarization. Here now we are talking about just membrane potential. So typically the values are between minus 80 millivolts to minus 40 millivolts. So I told you there is a buffer range, right? So this buffer range ends at minus 55 to minus 40. So there comes your threshold. So the typical values are this, depending on the different types of cells and different environment it is also. And how is it generated? Unequal distribution of ions, particularly potassium, sodium, and chloride ions across the plasma membrane. So, you know, there is influx and efflux of sodium, potassium, chloride, and ions. There are channels for it. Few of the ions just diffuse. There are some gated channels, there are voltage gated channels, sodium gated channels. So, all these are allowing your movement of the ions from from your plasma membrane from outside to inside or inside to outside, depending on the environment and the stimulus. What is the importance? The importance is, it is very crucial for proper functioning of your neural and muscular systems. So in your nervous system or your neural system, you know, it is important for your neurons for generation of your action potential. That is how the cell signals, and that is how your neurons send electrical impulses to others and communicate. And in your muscular system, you know, your muscles need to relax and contract, and then there also your action potential, your membrane potential, your polarization, depolarization takes place, especially like in cases like your heart muscle. It is highly uh, active muscle where it has to continuously contract and relax. So there also you get your ECG graph, right? So it is similar to your depolarization, polarization. Based on this only your muscle will also contract. Okay. So if you have any doubt at any point, you can ask me. You can turn on your mics and you can ask me. So the next question is, during repolarization, this is repolarization. During repolarization, membrane potential shifts from negative to positive, B positive to negative, C remains unchanged, D none of the above. What is the answer for this?
Ma'am, B positive to negative.
B positive to negative. Very good. So first in the previous one we saw what was depolarization. Then now we are saying what is repolarization. So your depolarization was from your negative to positive. So then it has to again come back, right, to resting membrane potential. Resting itself means your membrane potential will be always maintained in that point. So once it is depolarized or it is activated, then it should again come down. Only then it will go back to normal state, otherwise it will be continuously depolarized. So when it comes back, the membrane potential from your positive to negative, that is called as your repolarization. So during repolarization, the membrane potential shifts from a positive value to a negative value. This occurs after depolarization phase of an action potential when the membrane potential changes. Okay. So this is the similar graph. Here there are little more, there is little more information. So here also same, your minus 70 is your resting state or your resting membrane potential. Then at between your minus 70 to around minus 55 or minus 50 40, there will be your buffer zone where it will be continuously changing for different cells. And then there is a threshold. When this threshold barrier is broken and it further increases, then it attains depolarization. So this is your action potential. After that it will come back to the normal state, normal resting. So that will be your repolarization. In between this, there will also be certain failed initiations. So whenever there is a stimulus, it could be below the threshold. So if your stimulus did not break the threshold, or if it was not enough to go above the threshold level, then it will just go a little bit like that and then go back to the resting state. So this is called as failed initiations that you can see here. Then after repolarization also, it will go back further down sometimes. This is called as refractory period, because all this depolarization and repolarization are due to influx and efflux of your sodium and potassium ions. So if this during repolarization, if your influx or efflux is more to bring back your resting membrane potential to minus 70, then it goes into the refractive phase, where it goes further down than the resting phase, and then eventually it comes back and it neutralizes the membrane potential, that is maintaining your minus 70. Okay, so this is how your axial potential works. Next question. During depolarization, which ions move inside the cell from the outside? A. Sodium. B. Potassium. C. A and B both. And D. None of the above. That is neither A nor B. What is the answer for this? Which ion moves?
Ma'am, sodium.
Okay. So, during your depolarization, your ion which moves from outside of the cell to inside is sodium. Correct. Good. So, your answer for this is A. So, here you can see during your resting potential, there will be both influx and efflux of your sodium and your potassium in a balance. So it is, I told you, it is a dynamic process. So there might be a little changes. It's not exactly minus 70. Why that is? Because it's not like once the sodium is inside, it will always be inside, or potassium is outside, is always outside. It's not like that. So your cell, there is continuous flow of these ions. So it should maintain it in a balance of the concentration between the two ions. Then only the resting potential will be maintained. If this is altered, then you will know that there is some issue in the cell, or there is some activity that is going on. So that is what happens in case of depolarization. So in depolarization, there is increase in the movement of sodium. So that is what you can see with the bolder arrow. So when there is increase in the movement of sodium, the sodium levels will increase in the cell, and then because sodium is positively charged, so your minus 20 will slowly start becoming positive and positive and positive. So if there was not enough sodium ions to break the threshold barrier, then your depolarization will not occur. If it breaks, if your sodium is enough, good enough concentration to break that threshold, then it will increase to positive. So it will go to plus 30. So that time it will be depolarized. Then later what happens in the previous question as we spoke, it will repolarize. So that time what happened, there will be again efflux of this ions. So that time whatever positive ion was inside, that will be effluxed out, and then again it will be maintained in your minus 70. So this is how sodium and potassium play a role. So in your action potential here, you can see it's clearly explained. So this is your RMP, that is your resting membrane potential. Here instead of the line, they have shown this slant angle in your curve. So this point, up till this point, it is your threshold. So from your threshold, there is an increase, that is your depolarization. So this is because of the opening of voltage gated sodium channels. So your sodium influx will be more because the channels have opened and there is influx of sodium. Here it is saying that the local potential change, graded potential. So because of this balance that is in and out continuous. So because of that, this little change might be there, but then it won't par your threshold. Once it passes your threshold, then the depolarization will happen due to opening of your sodium channels and influx of sodium. Then after that, it has to come back down again. So then your repolarization will happen. So here you saw there was influx and now there is efflux. So your sodium channel will close and your potassium channel will open. So now it is both a play between your sodium and potassium. Then there is hyperpolarization or your refractory period, which you saw there. So that time your voltage gated potassium channels remain open and potential reaches resting level. So it can't exactly go back to minus 70 or minus 80, whatever the potential of that particular cell is. So sometimes it will go below that and then it will slowly come back to the actual potential. Okay, so this is how your sodium and potassium ions will play role. So here you can see in your neurons, your resting membrane potential is minus 70. So there is your threshold. This is your sodium channel. So now your sodium is entering your cell. This is your cell membrane. So you can see outside and inside you have positive and negative ions. The charge here is because of the ions. So when your sodium gated channel has opened, your sodium enters and then your charge inside your cell near your membrane will be positive now. So this is your depolarization. Then now you can see that sodium is inside and now your potassium is being sent out. So when your potassium is being sent out, both are positive ions. So your sodium will be now passed on. In your action potential, it has to send the signals, right? So you can see here, after the depolarization is done, the sodium will start moving, or the wherever the impulse has to be sent, it will go there. So that is called your propagation in case of your neurons. So now it has to send, right, one neuron to other neuron, or then your other neuron to your cell. How will it send the signal? This is how it will send your electrical impulse. So that is your propagation. After that, repolarization of this cell. So your potassium is being pumped out now at the same time. So both of them will decrease the membrane potential. Now your sodium and potassium pump are working together and your resting membrane potential is being maintained again in the cyclical balance. Okay, so this is how your depolarization and repolarization happens by the help of your sodium and potassium ions. Okay, the next question is, which units of the muscle cannot be used for force measurements? Your muscle, you measure force. Which unit of the muscle cannot be used for this purpose? A. Myotube. B. Myofiber. C. Myoblast. D. None of the above. What is the answer for this? Tell me. You can also guess if you don't know the answer, not an issue. Let me see, what do you think the answer for this is?
Myotube.
So the question is which one cannot be used? So your myotube, your myofiber, your myoblast. Okay. One of you has said myotube. Anybody else has any other answer? No? Okay. So the answer for this is myoblast. That is C. So now let's just see what is a myotube or a myofiber or a myoblast. Your first of all, there are different names for your muscle cells and fibers based on their arrangement and type of cells. So main is your myofibril, your myocyte, and your sarcomere. Okay, so your myofibril is a component of your muscle fiber or a myocyte that plays role in your muscle contraction. Okay, that is also called as your myotube. Okay, so now your myocyte will form groups of tissues and then it will become a myofiber. So a muscle fiber that contains thousands of myofibrils will become your myocyte. So you have fibrils or myocyte. These are sometimes, interchanged or used more or less as similar depending on the context of it. But if you want to be exact, then you have your muscle fibers and then you have your myocyte and then you have your sarcomere. Let's see the arrangement here so that it will be little better. So now what is your myoblast? Your myoblast is your, so you should recognize certain cells with the names they have. Blast, if anything is named blast, myoblast or some other osteoblast or something, it is a precursor cell or it is a progenitor cell. So it is a stem cell like your cell which is not yet defined to become a certain type of cell. So your myoblast, it is its fate has now been determined to become a muscle cell. One second. Sorry. So your myoblast is a progenitor cell or a precursor cell of your muscle cells. So you cannot actually name it a muscle cell because it has not yet acquired all the properties and it is not differentiated to be a muscle cell. So that is your precursor cell. So you cannot use a precursor cell to determine any characteristic feature of a differentiated cell. So that is why you cannot use myoblast. Whereas myotube or myofiber or myocytes, these are all parts of your differentiated muscle. So these cells can be used. So here you can see here, let me just go to the pointer. So this is your myocytes. Okay, your myofibril and then your myofiber. Your group of your myofibril will become your myofiber and then you have your sarcolemma and then satellites are external. So here you can see the arrangement. From your myoblast, you get your myocytes. From here, they become myotubes and then from here they become myofibers. So once they become, why this interchangeable terminology is there, because once they become myofibrils or myofibers, they cannot be really distinguished from each other. Now it's like a group of cells or a tissue. So that's the reason sometimes they are called in interchangeable names. But myoblast is completely a different cell which has not yet become a muscle cell. So it's your precursor cell. May okay, myotubes may coalesce and form few myofibrils. Imagine around 1,000 myotubes make 100 myofibril. So this is just to give you like appropriate or approximation of how many myotubes will become a myofibril, something like that. So your myotubes or your myocytes will become your myofibril. Your 1,000 myotubes make one make 100 myofibrils. So it's not exactly number kind, but it depends on the arrangement and how they are uh, coalesced, or how they form together. And then a bundle of 100 myofibril makes one myofiber or muscle fiber. So a fibril is a term similar to like a droplet. You have a drop, you have a droplet. So you have a myofiber, which is the actual bigger one, and then you have tinier versions of it, which is called as myofibril. So your 100 myofibrils will make your myofiber, and then it has a sheath covering called as sarcolemma. So this sarcomere or sarcolemma that will come around it. Okay, then many such myofibers together. Now this entire thing is your myofiber. Many such myofibers together with the help of endomysium as connective tissue. So to hold all this, you need a connective tissue. You can't just arrange the myofibers on one on top of the other. It won't attach. So you need a connective tissue to hold all this in that particular shape and position. Okay, so that is your endomysium, that is your connective tissue. And make a muscle fascia. So your fascia is a big portion of your muscle, which is made of hundreds of many muscle fibers. Okay, so these fascia, you might have heard terms like fasciatic pain, when there is pain in just one particular region of your muscle. So that is your fascia, with which in turn, with the help of perimysium, this is another connective tissue, because it's peri, it's outside. When it's inside, before it was endo. Okay, so you should know the terminologies, why they are termed, so that you can remember easily. And the covering is called as epimysium. Epimysium is outermost to the entire thing. So first you have endo inside, peri above that, and then epi outermost. This is what makes up your one muscle. Okay, I hope it's clear. It is a little confusing, but if you need more information on this, I can share some websites or URLs. Okay. So the next question is, neuromuscular junction is made at junction of which, it is made at which junction, between what? So your first is A, muscle and motor neuron, B, glia and motor neuron, C, hippocampal neurons and osteoblasts. See, again there is osteoblasts. So this is also a precursor cell for osteoclasts. Osteoclasts are your bone cells, osteoblasts are your precursor cells, and none of the above. So what is the answer for this? Neuromuscular junction, where do you think is it present? A, B, C, or D? Come on, tell me, somebody guess.
Mm.
Sorry, can you repeat it? I just heard.
Muscles and muscle.
Okay, I think you're saying muscle. Your voice is not very clear. Okay,
Option A.
yeah, very good. Option A. So your neuromuscular junction is between your muscle and your motor neuron. What is a require? What is a junction? First of all, something that connects one to the another. So this junction, neuromuscular junction or NMJ, is a connection between your muscle and your motor neurons. So it is responsible for converting electrical impulses from the nerve into an action potential in the muscle fiber. Now, before we saw the electrical impulses that was happening in the neurons. So this neurons have to now stimulate something. That is why they have this electrical impulses. So how do they stimulate some other tissue or organ to convert it into some kind of function? So that is how they convert the electrical impulses into uh, action potential in case of your muscle fibers, because your muscles have to now contract and relax. So that also happens because of your action potential, where the depolarization repolarization will now also occur. Sometimes in other types of cell, if there is some signaling that has to happen, so that time your electrical impulse will be converted into some sort of chemical signaling, like some release of some kind of uh, prostaglandins or some kind of hormones or some kind of messengers like calcium, etc. So this is how your junctions work. Then it also includes a synaptic end bulb, motor end plate, and synaptic cleft. So these are all part of it. The cleft has to be there, the end plate has to be there to disperse it. The motor end plate is a region of the muscle fiber that contains thousands of receptors that bind to acetylcholine. So now, just now I was saying about some chemical impulses that it will be converted, like prostaglandins and all. So acetylcholine is also a kind of chemical that is released when certain kind of action potential or certain kind of electrical impulse is generated by the brain. So your acetylcholine will be released, and then your target region should have the receptors for it. So your acetylcholine will go and bind to the receptors, and then downstream your receptor, certain phosphorylation or some kind of other modifications will happen, and signaling will continue. That is how your electrical impulse is converted into whatever function it was intended to. So this is very important in many of the diseases. So when there is uh, a problem or a dysfunction in your NMJ, that is your neuromuscular junction, uh, there are it leads to many muscular disorders or muscular dysfunction, like in case of myasthenia gravis or your Lambert-Eaton syndrome, botulism, and also cause muscle weakness and paralysis. Because if this is impaired, then your signal will not be transmitted, or your muscle may not be capable of receiving the signal and converting into chemical signal, and then your relaxation and contraction will never happen, even though your brain is sending signals, or you want to do certain movements. So that is the reason for kind of paralysis and muscle weakness and other muscle disorders. So this is your neuromuscular junction. This is how it looks. You can see so many molecules are released because of the uh, electrical impulse that was transmitted. So that is the end of your axon. That's your axon terminus. And then there is your myelin sheath. Your axon motor neuron will have its connection to your this muscle at the other end. So you see, this is your muscle fiber. It has many receptors. It has your neurotransmitter gated channels, and then there is you can see that there is a labeling of a motor end plate. This is what they were talking about. And then these small molecules that are released are your acetylcholine that is released. And then this is your receptor, which is your acetylcholine receptor. You can see it will come and interact with your receptor. See here, it was closed. Now when the uh, acetylcholine has interacted, now your gate has opened, and then you have your acetylcholine esterases. So this will allow the entry, uh, further downstream activity of this. And then this action potential will now propagate from here. In the previous one, we also spoke about propagation of the action potential, that once it depolarizes, then it has to propagate that signal. After that, it will repolarize. So here you can see that once the signal is getting activated, again your sodium and potassium are playing role because it is an action potential. Depolarization has to happen, and now there is propagation. You can see now this will continue. It started over here, it's going on, then it will continue to the other fibers surrounding it. So that is how the entire region will get affected. Okay, so this is your voltage gated sodium channel, and then this is an actual like an electron micrograph or image of your axon terminals. So this is your muscle fiber, this is your neuron, the darker region like a silhouette you can see, that is your neuron with branches. And then you can see this end plate that is attached to your muscle fibers. Okay, so the terminal button it's also called, so your axon terminals attached there with your muscle fibers, and then the signal will be propagated like this continuously throughout the length of your muscle fiber. Okay, so that is how then your muscle fiber will start to contract or relax, convert this electrical impulse into chemical impulse, and then a physical movement, mainly because it's a muscle. Okay, hope this is clear. Next question is Vagarotoxin causes paralysis by blocking one of the following. What is that? A. Potassium channel. B. Acetylcholine receptor. C. Both A and B. And D. None of the above. What is the answer for this? Which one does
Both
Vagarotoxin?
B.
Yeah, can you repeat it again? Sorry, it was not audible.
Both A and B.
Yeah, both A and B. Very good. So your toxin. Sorry, I was supposed to write both A and B. Okay. So your it's both. Let me just write it here. Okay, so this is your option C, both A and B. So your toxin again has forms like alpha, K toxin, etc. So your alpha and K toxins are usually from your snake bite venom. So it's found in many of the banded crates that are found in different parts of Asia. So the alpha toxin binds to the nicotinic acetylcholine receptors at the neuromuscular junction, just now which we were talking about, and it can be used to detect anti-acetylcholine receptor antibodies. And then your K toxin also binds to your acetylcholine receptors, but in many of the cases, the toxin is also known to block your potassium channels as well. Okay, so this is like how it basically happens. So you have many things going on here. You have, where is the cursor? Okay, laser pointer. Okay, so you can see here you have both. You have your potassium channel and you have your acetylcholine receptors. So this will affect both the voltage gated potassium channels and your acetylcholine receptors. And not only this, uh, your bagarotoxin, it also shows varied effects. So it is not just that it will block it, but it will also show different type of effects, like it might block the receptor partially sometimes, it might block the receptor completely, or it might, sometimes enhance it also. There are certain reports which also suggest that. So your toxin has, it's known as a triple effect of your toxins. So there what happens, so sometimes your acetylcholine, your function will be either partially blocked or completely blocked, or sometimes it might also be enhanced. So it is used basically in many of the neuron related research. Okay, so the answer for this is C, not A. Okay, it's both A and B, it's C. Okay, next question. For myotubes cultured on cantilever devices, to move cantilevers, which property of cantilevers should be similar to the myotubes. So cantilever is something on which you can grow your myotubes, and it's part of an instrument, and it is then used to measure some property of your different properties of your myotubes or your tissue, muscle tissue. So the it basically functions by deflecting the light. So your this cantilever, wherever you are plating, so it will move, and it's a rigid structure, but sometimes it will, bend, or it will angle in order to deflect the light. Based on that, we can measure. So what property of this cantilever should be similar to your myotubes for this measurement to be accurate? Is it A, stiffness? Is it B, color? Is it C, weight? Or is it D, width? What is the answer for this? Anybody? You can guess anything. Think, based on the explanation that I have given about the cantilever, that it is a plate sort of thing of the instrument where you culture your cells, and then this is used to angle or to move in order to deflect the incoming light. By measuring the change in deflection or angle of deflection, you will then measure a property of your myotubes or your muscle fiber. So what, in such kind of a case, what property of that cantilever should be similar to your myotubes? Anybody? Can someone answer yes, no, A, B, C? Anything? Are you guys there? Come on. Yeah, I can see few people. One of you can answer it, at least guess something. Anisha, Krishna, Kumaran, Pranab, Soumya. Come on, guys, one of you tell me what is the answer for it. Myotubes cultured on cantilever. What kind of cantilever is appropriate for proper measurement? Should it have similar color, similar weight? Yeah, yeah, tell me.
Option A.
A, stiffness. Yes, very good. So, your cantilevers, like I said, it's a part of a device or a instrument. So, it can be used to study various properties of your myotubes. It can monitor the deflection. That is how it works. The vertical deflection of the cantilever can be monitored in real time. So, it is a time based, like immediately you can measure based on your stimulus or treatment how it changes. Okay, like real time PCR, you can measure this in present with time as a function. So it can also measure the contraction force of your muscle. So the force that is present in the muscles can be measured while contraction. It's also called like shear force or tensile force of your cells. It can also quantify the contraction fidelity. the fidelity of the muscle upon contraction in each stimuli can be quantified. Not only it can measure it, it can, give a quantitative analysis about it, how accurate or how you know efficient it is. Okay, that is your contraction fidelity. Then investigating physiology and kinetics. So your kinetics, like your uh, movement, your muscle, your action potential, which one is more efficient, these kind of things can also be measured. Then testing of the effects of toxins. Effects of toxins on contractile function of myotubes can be monitored. Like I said, like different treatments with it, like you can add some kind of toxin on it, like this toxin or something. You can see what concentration is affecting, which concentration is more, toxic, and what level is bearable for the cells. So all these kind of things can be also tested. Then investigating the effects of medium composition. The effect of composition of the medium on the contractile ability of myotubes. So your myotubes also require certain, nutrients and specific components in the media for its proper growth and function. So because they are very specified cells, like most of the differentiated cells with a particular property or a very specialized property will need certain essential elements in their media while culturing, that are required for those particular type of cells to efficiently function. So in the same way, you can, based on, because there will always be a standard after a culture has been created and maintained, or standardized for a particular in vitro growth and culture. There will always be standard for every type of cell, that certain cells when differentiated should show certain properties, and certain cells when differentiated should show certain functions. So in many cases these properties are, measured by several um, protein markers. Like if there are immune cells, you measure some kind of CD markers, cell surface markers on them. If there are some kidney cells, you measure some kidney specific proteins. If they are muscle cells, you can also measure muscle related proteins. But the most efficient way is to measure its ability, because your main function of your myotubes or your muscle fibers is to contract and relax. So if this ability is functional in the cell, you can use this, that as a proper standard cell line. So that can also, or maybe if you're generating a new muscle cell line using some other source, so then you can compare its contractile ability with other muscles. So for this also you can check if there is any other requirement in the media that is, you know really required for it to function properly. So this is how the device looks. So you can see here there are pre-differentiated cells. They look different. Their arrangement, you can see it's like a normal epithelial monolayer of cells. But then once they have differentiated, they have rolled up and they have arranged nicely like a myotube or a muscle fiber or a myofibril, okay, based on the size and number of it. So here you can see this is your cantilever. You can see how this is movable. Every cantilever, it will have a certain level of rigidness. So based on that, you have to use your culture, or you have to, buy appropriate stiffness in your cantilevers according to your myotubules. So here you can see this is around 100 microns in width, and around uh, you know 1000 or 700 microns in length. So, and its thickness is around 3 to 4 microns. You can see this is your light that you're talking about. This is your laser. then the laser will get deflected by your cantilever. So this can also help in moving your uh, sample, okay, moving your sample above or below, or in y direction mostly, or your x direction, anyway, according to the device. So here you can see when the laser hits the sample, it has been deflected to a certain angle or a certain uh, arc. So now this deflection will be detected by your photo detector, and now your photo detector will collect the information and it will give you this as a signal or an image. So here you can see this is the top view. This is your cantilever. You can see that there are myofibrils on this. You can see very thin, I'm not sure if you're able to see it. You can see very thin filaments on it which are grown. So that is how it looks. This is your top view, and this is your side view. This is fluorescently labeled so that it will be visible to the naked eye, and from the side view this is the thickness of it. So here you can see healthy engineered muscle. So this is your healthy engineered muscle. This is how the signal will appear after collection from your cantilever in your photo detector. So you can see these are, it gives you different signal based on the surface and in your length and breadth and width and various kinds of information, and how much contraction is there, etc., can also be measured using this. Okay. uh, Next is hippocampus. I think this has already been discussed before. So your hippocampus, is it associated? What is the function of it? Is it associated with A, movement, B, color vision, C, learning and memory, D, vocation? So what is the answer? Hippocampus. This was also discussed in the very beginning sessions. What is the function of hippocampus? Tell me. Yes? Anyone?
Option C.
Option C, learning and memory. Yes. So this is your basic structures of your brain. So these are your smaller structures. You have your basic cerebrum and cerebellum, and then this is your basal ganglia, which controls the movements, learning, habit, cognition, emotion, etc. This is your thalamus behind. It regulates your sleep, your consciousness, your alertness, etc. Then you have your amygdala, which is mostly for your decision making and emotional responses. It is also part of your memory, like your decision based memory, or your previous whatever you have done. And then your hypothalamus, below your thalamus, it controls your body temperature, hunger, fatigue, sleep. Your basic body homeostasis is is maintained by your hypothalamus. And then you have your hippocampus. So your hippocampus is also for your memory and navigation. So your basal ganglia, whatever you're seeing here, so here you also have this hippocampus, your C like structure over here. So what is hippocampus? Hippocampus means it is the seahorse like structure. So that is the reason it was named hippocampus. Okay, it's called as a seahorse monster, something like that. So the hippocampus is the main lobe that is responsible for learning in an individual. The lobe is also responsible for major areas of memory, with one study concluding that hippocampus is responsible for organizing memories into a timeline, and another concluding that it is involved in both short term and long term. So your chronology, you remember that so and so thing has happened in so and so time, and all this timeline dependent and long term, short term memory, everything is controlled by your hippocampus. So there are some other facts like your hippocampus is part of your temporal lobe and your limbic system, and it looks like a seahorse. It all mammals have two hippocampi. Okay, these are your basic things. Next question. Dash is a pungent pain producing compound found in plants of capsicum family. It exerts excitatory, desensitizing, and toxic effects on a subset of sensory neurons, including polymodal nociceptor population. Is it A?
Capsaicin.
Yeah, okay.
Option A, capsaicin.
Very good. Now you're alert. So the answer is A, capsaicin. So with the name, you can easily recognize capsicum, capsaicin. What is capsaicin? Capsaicin is a fat soluble phenolic compound that gives chili peppers their characteristic hot and pungent taste. So the capsaicin interacts with your pain and heat sensing neurons. So your we say the taste is hot or spicy or pungent. So all your capsicum, your chilies, not only your capsicum family includes all types of chilies, red chilies, green chilies, everything. So they interact with your heat sensing and pain. So pain because it's spicy, you feel the pain, and then heat because heat sensing neurons. So then it also generates lot of heat. So you feel like it's very, your temperature is getting raised, and it causes a burning sensation when it comes in contact with the mucous membranes. So wherever there is mucus related part, like your saliva, also your tongue or your esophagus lining. So that's why a lot of times if you eat very highly spicy food, your burning sensation continues even through your esophagus. So why this is important is it is very medicinally valuable compound, and it is very useful in treating or at least managing many types of cancers also. So here I have given few examples. So in basic internal medicine, it is used in treatment of many cardiovascular diseases where it improves lipid metabolism, and then it lowers the blood pressure by diuresis and natriuresis stimulation, and then it increases energy metabolism. And then in chronic kidney disease, it decreases the inflammation. So if there is like injury within your kidney cells or there is inflammation, so this continuous chronic inflammation will also be decreased by this compound, and it decreases the oxidative stress. So your ROS, your ROS production, all this reactive oxygen species, your superoxides, they create this kind of oxidative stress to your cells. So this kind of cells will also have a relief from this because it's rich in antioxidants as well. So this chilies, peppers, all these are they eliminate the bad oxygen or your reactive oxygen species, which in turn will also decrease your inflammatory state. And then it has nephroprotective action. Because of all this, it protects your kidneys, and then it decreases the blood pressure by diuresis and natriuresis. So your blood pressure is not only a issue with your heart, it's also your issue with your kidneys, because in the kidneys you have your nephrons, and the pressure that has with which it has to be you know passed into your nephron, your glomerulus. So it has a particular pressure called as, there it is called as glomerular filtration rate, the pressure which due to the pressure of your blood. So this rate has to be maintained so that your blood has been effectively purified and your toxic is released in your urine. If this blood pressure is not maintained, it will in turn affect your glomerular filtration rate, and then in turn decrease your filtration capacity, and your toxins will not be eliminated, or your important things from your blood will be eliminated into the urine, and certain important proteins like albumin, C-reactive proteins, etc. All these also will be lost into the urine, and it will cause a condition called proteinuria, which is a basic for all the kidney related things. Next, it is also helpful in metabolic diseases or metabolic, disorders. Here, particularly in case of diabetes related and obesity related, so which are more or less again similar. Because of diabetes and obesities, there are secondary complications that occur. It's not just that you have diabetes, you have problems. How the problem arises is the secondary complication that comes. It gives a load on your kidneys, it gives a load on your heart, it gives a load on your liver. So because of your this obesity and diabetic condition, your vital organs will have complications. So it is called as diabetes related secondary complication and obesity related secondary complication. So in such cases, it helps in increase of energy metabolism in case of obesity, and thereby decreasing the body weight. In case of diabetes, it decreases the insulin resistance. These days there is a lot of increase in insulin resistance, that is your type 2 diabetes. And then it decreases your blood fasting glucose, and also increases your energy metabolism here. And not only here, it is also used in your dentistry, that is for your oral health. It improvement of burning mouth syndrome, post traumatic trigeminal neuralgia, your atypical odontalgia, trigeminal neuralgia, increased flow rate. So as it was said that it has both pain and desensitizing effect, so it is also involved in many of the pain management cases. In case of cancer patients or some other patients, it is also used to decrease the pain, or desensitize the pain receptors. Because how can you decrease the pain? It's your painkillers. What do? They desensitize your receptors. That's how you don't feel the pain. So this also has this kind of property. A last question. Dash blocks sodium channel. A TTX, B 4AP, C calcium, D free radicals. What is the answer for this? Sodium channel blocker.
Then C option.
C calcium. You think calcium will block the sodium channel? Okay. Anybody else has any other answer? Yeah? No? Okay. So the answer for this is A, TTX. Your TTX is a tetrodotoxin. Okay, that is the short form TTX, tetrodotoxin. What is it? It is a neuronal toxic uh, chemical substance. So it is produced by many biological uh, life, like your marine life, mostly your puffer fishes, your snails, your octopus, and many other reptiles also. So your TTX, your tetrodotoxin, is a low molecular neurotoxin. So it's easily, you know, it can easily go and spread at different parts because of its very low molecular weight. That it can, circumpass some barriers. So the how it does is it blocks the pore region of your voltage gate and sodium channel. How it blocks the channel? It will block the pore. So your gate will have a pore in the middle. through which your sodium will enter. But instead of that, this will go and block the pore region. It prevents propagation of your action potential. Obviously, if your sodium channel is blocked, how much of a stimuli you guess, your channel will not open, or your channel will not allow entry to the sodium. If the sodium levels does not increase, your depolarization will not occur, and then there will be no action potential. So there will be death due to asphyxiation. So asphyxiation is your suffocation. So your lungs also have to contract and expand and contract, right? And then your nose, when you inhale, it has to go and reach your lungs. So if there is this kind of no contraction and relaxation happening, so the first thing that will get affected is your lungs. It will not get enough oxygen, and then you will have suffocation. So that is how this tetrodotoxin works. You cannot breathe. It is found in variety of marine animals. So this is your sodium channel, your voltage gated sodium channel. So you can see it's a tetramer. This is one protein, this is one protein, this is one protein. It's a tetrameric protein, and this is the arrangement of it. And this is your pore. You can see a hole in the center of it. It's like a cylinder, and then at the center there is a hole. So it's a pore through which your sodium will enter the channel. So this will be on the outside, and this is your inside of the region. If you draw a line like this is the outside of your cell surface, this is inside. So your sodium will enter like this. So you can see here there is like several structural details. You can see there is a voltage sensor, there is a selectivity filter that it has to, it is specifically for sodium. So that means it should not allow any other type of ions. That's why it's called as sodium channel, otherwise it will be called as an ion channel. So it has a selectivity filter which will allow only sodium. And then you see this ball sort of region. This is the region where your TTX comes and binds. So it's exactly at the center of your pore. So if it comes and binds, it's present on all the subunits, all the four uh, subunits of your protein, or all the four monomers of your protein. So if it comes and binds in this region, your entire pore will be blocked. So this is your TTX binding site. How does it work? It works by blocking the flow of sodium ions through the channel, preventing the generation and propagation of action potential. Where it is found in variety of organisms like pufferfish, newts, gastropods, crabs, and starfishes. What are the symptoms? The symptoms are TTX poisoning includes respiratory failure. Like I said here, asphyxiation, it's your suffocation and you cannot get enough oxygen or you cannot breathe. So first what happens is your respiratory failure, and then hypotension, low blood pressure because there is not enough oxygen, and then weakening of voluntary muscles. Then eventually, because it's preventing action potential, it will stop movement of your muscles as well, and then there will be no voluntary movement. Then what is the use of this? TTX is used as a research tool to study voltage dependent sodium channels. It is also being explored as a treatment for some types of pain. So because it binds and blocks certain channels, so it can also be used in blocking pain receptors, or maybe some other way where the it might stop the action potential at a particular site where the pain is. So it is used in several research. Okay, so hope this was helpful and useful. The questions are done. If you guys have any questions in any sorry, any doubts in any questions, you can ask me. I think we have a lot of time left.
Ma'am, is it the last live session of this course?
Yes, this is the last live session, but there might be an extra session. I have mailed regarding it. So there could be an extra session. You will be informed. But official, like according to the timetable, this is the last session.
Okay.
Okay. Anybody has any questions?
Ma'am, can you suggest anything that how should we prepare for the exam?
How should you prepare for the exam? So basically, the main exam will be conducted by the professors and the NPTEL team. So I'm not really aware of the pattern of it, only the assignment pattern and the theory, the concept, the what is it? Your syllabus is known to me. You can do one thing. You can put up a query in your group. There is your NPTEL group, right, where you get the announcements. So you can put up a query there. Someone from the support team will be able to answer you.
Okay.
And mostly, if you want like in sort of preparation that is regarding your exam pattern, in case of preparation, you have your basic notes. So you can first go through your notes and then your videos, and mostly it will come from the notes and videos only. If you think that was not enough, then you can go into certain uh, you know you can refer some books and all. If you need some references, we can provide you. You can put up a query for references in your support group, and we can give you some references for it, some books or some websites.
Okay.
Okay. Anything else? Okay, since we have some time left and you guys do not have any questions, I just wanted to go through the do's and don'ts of cell culture because this is a cell culture technologies. I had done this in one of the earlier sessions. So, since it's the last session, I just wanted to go through the basics which will help you in your practical uh, work when you start your cell culture. Okay, so first is your do's, then your don'ts. So first one, we'll go through it one by one. Use personal protective equipment, lab coat or gown, gloves and eye protection at all times. In addition, thermally insulated gloves, full face visor, splash proof apron should be worn when handling liquid nitrogen. So, in most of the chemistry labs, they already do this, but it is also important in cell culture labs because sometimes you use chemicals, for disinfection or for storage purposes. So, those times you need these kind of protection to protect yourself, your clothes, your skin from getting burnt. Okay, so you need to use gloves first and foremost. Gloves is for your own protection and also to avoid contamination in your cell culture. Wash your hands neatly and then dry them properly. Don't wear gloves on wet hands because that will again be contamination. So after that, wear your gloves and then clean your gloves with ethanol so that you will kill whatever bacteria or fungus or anything that is present even on your gloves. Once your ethanol is dry, then you can start working. Your thermally insulated gloves is for your storage purposes, like when you store something in liquid nitrogen, the temperature is in minus, right? So it's minus 130 something. And then when you store your samples in your minus 80 refrigerators, so that time you get your frost bites, like how you get burnt with very hot boiling water, you also get burnt with negative temperatures and negative temperature ice. So you have to wear in the thermally insulated gloves, full face visor, and other things are mainly for when you're dealing with sort of uh, hazardous or some pathogenic viruses or bacteria. So your entire even you in highly uh, pathogenic labs like BSL facilities, high BSL facilities, they also use the PPE suits. So this is very important. First, protection for yourself, and then avoid contamination. Always use disposable caps to cover your hair. So your hair is a very good source for contamination. You can get, uh, you know, when you travel, your dust and all settles on your hair, or your scalp. Many of us have dandruff. So when your scalp, your dandruff is nothing but fungus, and then dry. It might just spread. You wouldn't even know that it is being spread, but it might. So cover your hair with caps, and wear dedicated PPE for tissue culture facility, and keep separate from PPE worn in general lab environment, your PPE, your suit or your lab coat or whatever. So you can also do this by wearing different colored coats, or having a name tag on it that this is only for cell culture. So something which you do in cell culture, you should not use that outside in your general laboratory. If it's for cell culture, it should only be worn and kept in the facility of your cell culture. Using it inside and outside will only cause contamination. Then there is no point of even wearing it. Okay. Your next is keep all work surfaces free of clutter. So mainly your laminar airflow, your hood, and your cell culture facility, your tables. If you're placing anything, only keep things that are really really important. Do not keep anything that is unnecessary should not be kept, and especially your laminar airflow should not be cluttered. It should not have so many things that the air doesn't pass, your sterile air doesn't pass through all the area. Or your UV lamp should not be you know should be accessible. Your UV light should be accessible at all points in your laminar hood. You should not obstruct it, otherwise sterilization process will not be efficient. Correctly label reagents including flasks, medium, and ampules with contents and date of preparation. So labeling is also very important, because you label something today, and then if you want it after few months or maybe even few years, you should know what the content is exactly, not just the name, the concentration of it, the date of it, and if any other important information that would be required for reuse of it. So if you don't label, you might think that I will remember this tube that I have kept it in so and so point. But then after a few days, you will not remember, but because you have done so much of work, you will not remember if it is yours or somebody else has come. Mostly these are shared environments, right? So someone else has come and put their tubes. They also didn't label. Everything looks same. The liquid looks same. You don't know which one is yours. So you have to discard it. So most of the reagents that we use are very expensive. So avoid wastage. We can always label these things, and also to avoid confusion and be sure of what you're using. And date of preparation is very important, because you should know how old your reagent is that you're using. Only handle one cell line at a time. This common sense point will reduce the possibility of cross contamination by mislabeling, etc. It will also reduce the spread of bacteria and mycoplasma by generation of aerosols across numerous opened media bottles and flasks in the cabinet. So usually when you do more than one cells in one incubator or one cell culture, if you do not label it, you do not know which cell is what unless you revive it. Even to revive, sometimes you need a different media. So labeling is very important. And then if you avoid using two or more cells in the same incubator, if you can use one incubator to one cell line, then you will avoid lot of contamination. You will avoid lot of confusion or mislabeling sorts of incidents. And then spread of bacteria and mycoplasma, these cannot be eliminated once they have attacked your cells. You have to only discard your plates. So you should be very careful not to get your cells contaminated by mycoplasma or bacteria. And bacteria, they produce spores. Fungus also produce spores. So these can spread through air also. You might think there is only one plate which is contaminated, the other plates might be fine. But if the contamination has been prolonged for more days, then it will, if it's a spore producing organism, the spore will spread across your incubator, and then other plates also will get contaminated, and this will affect the reproducibility of your results. So once you have done some experiment, you got a result, but then next time you did everything the same but you did not get the result. So if there is contamination, you will not get same result again. Okay. So also, when you open your media bottles or flasks in your cabinet, never leave them opened. Always close them immediately. Most of the times open them within the uh, you know laminar airflow itself. Do not open anything outside. Open it in a sterile environment and close them properly, tightly seal them. Next, clean the work surfaces with a suitable disinfectant. Example, 70% ethanol between operations, and allow a minimum of 15 minutes between handling different cell lines. So even if your incubators are different, if you're using the same hood for two different cell lines, after, do not split or culture or seed two different cell plates together. Always finish one cell plate first, and then clean the laminar airflow neatly, and then turn on the UV for 15 minutes, and then go for the second type of cell. This will help you in avoiding contamination or cross contamination within your cells also. And be very careful while using your tips and tubes and your serological pipettes. When you're using, uh, it should not be mixed up in two different cells. Okay. Wherever possible, maintain a separate bottle for media of each cell line in cultivation. So even if there are 2 3 people working, it's always better if each person has their own media. Do not share your media bottles. And if you're doing two different types of cells, obviously there should be two different bottles for each cell line, even though you use the same media for both. For example, if you're using DMEM for both the cells, do not use the DMEM from the same bottle. You should use one, you should label it, this is for HEK, this media bottle is for HG, something like that. Okay. So do not mix up your media bottles, and name your name. Do not share it with others. Even when using, transfer the media from the main bottle, your 1 liter bottle, to your 50 ml tubes, and then use from your 50 ml, pipe it out from your 50 ml tube, so that your main bottle does not get contaminated. Examine cultures and media daily for evidence of gross bacterial and fungal contamination. So every day you have to check your plates in the microscope and check if there is any contamination, to make sure that your cells are healthy and in the proper morphology and function. This includes medium that has been purchased commercially as well. So it's usually, this is done when you prepare your media. You can prepare your media from scratch in the lab, but mostly when media is commercially purchased, sometimes we'll be sure that they are sterile because it's certified. But still, you have to always have your own quality check. And if you have a doubt, just put a little bit of media in a tiny 35 plate and put it in the incubator for 2 3 days if there is any sort of contamination, because when it's in 4 degrees you will not see any growth, even if it is contaminated. So when you put it in a plate and put it in the incubator, just the media, then at 37 you will see if there is any contamination, it will start growing, then you can immediately discard that bottle. Okay. Then quality control of all media and reagents prior to use. So reagent bottles, your media, everything should be sterile and properly handled in sterile environment. You have some reagents that you add in your media after purchasing, like your antibiotics or your FBS. So you should take maximum care when add these reagents into your media. Keep cardboard packaging to a minimum in cell culture area. So all the cardboards, in cartons in which you receive your cell culture plates or your tissues or your reagents. Do not put, it's better not to put the cardboard boxes inside, because your cardboard boxes have traveled from out and it will have collected dust already. Even if you spray ethanol and anything, it is wood, it's made of wood, so it will collect more dust, and sometimes if it was in a transported in an unhygienic environment, it might be a source for your contamination. So always take out, you might have cabinets in your cell culture, so take out the things from it and put the individual packets in your cabinets. If not, put the carton outside, and whenever required, take the things from it and spray it with ethanol nicely, wipe it, sterilize it, and take it inside the facility. Ensure that incubators, cabinets, centrifuges, and microscopes are cleaned and serviced at regular intervals. Test cells for mycoplasma on a regular basis. So your all your things that is in your cell culture facility, your incubators, your cabinets, your centrifuges, your microscope, your refrigerators, all these things should be you know periodically cleaned, sprayed with ethanol, and serviced regularly, so that there should not be any contamination from it, and it should work efficiently so that your cells will not have any kind of stress. And mycoplasma is contamination that cannot be detected most of the times. So there will be a kit available. You can always check if your cells, if you have received it from an unreliable source, or if you have some kind of doubt, you can always buy the kit and check it for mycoplasma contamination. Okay, these were the do's. Now we'll go to the don'ts in the cell culture. So do not use antibiotics continuously in the medium, as this will inevitably lead to appearance of antibiotic resistant strains and may render a cell line useless for commercial purposes. So most of the times we use antibiotics, but sometimes you should also try to avoid it in certain passages or certain number of times. Only use it when it's a highly risk risky cell line, or if there is very frequent contamination. And even after antibiotics, if you are continuously getting contamination, that means you have generated an antibiotic resistant strain, which will now not be eliminated or cannot be eliminated. So you have to freshly again revive your cells, and maybe for the starting time you can avoid the antibiotics to check if it is contaminated or not. Next, don't allow waste to accumulate, particularly within the biological safety cabinet or in the incubators. So if you do not, if you're using some plates, or if you don't want to use the plates, if you have missed the date of experiment, do not keep the plates in the incubator for a long time. If you're not using it, immediately discard the plates, and do not accumulate waste. The the wrappers in which the plates come, or your tissue paper, anything that has to go into waste, immediately throw it in the dustbin. Keep a separate dustbin for the cell culture, and then immediately empty the dustbin often, so that the discard will not accumulate inside. Don't have too many people in the lab at any one time. So usually when you go into a cell culture facility, you have to go by yourself or one person extra. You should not go as a group. You should not have too many people together at one time. And also particular facilities should be accessed by only few people, to avoid more sources for contamination. So as soon as you enter the cell culture, there is a risk of contamination. So if more number of people enter, there will be much more risk of contamination. Don't handle cells from unauthenticated sources in the main cell culture suite. They should be handled in quarantine until quality control checks are complete. So usually when you're starting the lab, when you're buying your cultures, or even if you're buying some new cells, or you have taken cells from someone else, some other lab has gifted you the cells, you have your own cell culture line running. Do not immediately come and you know start culturing these cells along with your cells. Never, never do that. Always start, revive it, culture it in a separate incubator, preferably in a separate cell culture. And then for a couple of weeks, it has to be checked regularly to see if there is any contamination or if there is any other kind of problem or if the cells are healthy. Only when you are sure that these cells obtained from another source are healthy, free of contamination, then you can get it in your main facility. Avoid keeping cell lines continually in culture without returning to frozen stock. So every cell culture has a limited range of passages that it can undergo. You can subculture it so many number of times. So you have to respect that number of passages that is assigned for that cell culture. So typically after 10 passages, your cells start losing their morphology or they go into senescence. They do not divide very quickly. They are stressed. So that means you have over passaged them. So culture it, passage it to a particular number of subcultures and then freeze the stocks. Freeze it in your second, third passage itself so that the cells are young and healthy. So by the time you are about to reach your 10th or ninth passage, revive a fresh stock and then stop your previous stock at 10th passage. So that is how you maintain healthy cells continuously. This will also help in reproducibility of your results. Avoid cell culture becoming fully confluent. Always subculture at 70 to 80% confluency or as advised on EACC cell culture data sheet. So if you're purchasing a cells from a commercial vendor, they will have all these details: number of passages, required media, number of cells to be seeded, uh, confluency, maximum confluency to be maintained. So all these information is very important and you have to go through the data sheet first. So to any given cell culture, you have to always maintain it to grow it to a maximum of 80% confluency. So your confluency is the percentage of cells that are covering your plate. So you physically can't count each and every cell on your plate. So that's why you see a percentage of cells on the plate. If you can see half in your plate, half of the area is covered by your cells and half empty spaces are there. It won't be exactly half half, right? So your cells will be here and there, and in between you have gaps. So you should have an approximation and estimation in your brain that, like, there are some, this much area is gap, this much area is covered by cells. So if 50-50% is there, that means it is your cells are 50% confluence. So if your plate is maximum covered by your cells and there are few gaps here and there, that means it is closer to 80-90% confluency. If your plate is completely covered with cells, you cannot see the gap, you cannot see the surface of the plate at all, there is no gap, that means they are 100% confluent and they are overgrown. Never grow cells like this. You have to always grow them on a mono layer, that means a single layer of cells. Why we should not grow or we should not leave it to get over confluent is, if you grow more than 80%, then the cells will start overlapping on each other. So that time your cells will have stress. The cell which is under another cell will be in stress. These cells are standardized to grow in monolayers. Okay. So that is why always stop or subculture your plate when they are 80% confluent, or use it for experiment when they are 80% or below 80% for confluent. Again, for different experiments you will need different percentage of confluency. For western blot, RNA, etc., you will need around 70 to 80% confluent cells. For microscopic experiments, like if you want to use confocal, you want to do immunofluorescence, etc., then you will use less than 70%. You will use around 60 to 70% because you have to see at that high resolution, you have to see the individual cells. If they are overcrowded, if they are attached to each other, they will not even spread to their maximal capacity. They will be very close to each other, tiny places, not able to grow, no morphology will be visible. So all these things should be avoided. Do not allow media to go out of date. Shelf life is only 6 weeks. at at plus 4 degrees once glutamine and serum is added. So your media bottle, like I said, commercially it will come with a particular composition. Then you need to add your serum, that is, excuse me, that is your FBS. You have to add it after receiving, and your antibiotics or any other chemicals, or vitamins or glutamine. If usually glutamine will be present if you, in certain experiments, if you want to have different concentrations of glutamine or different concentrations of glucose you want to check, all these things you have to add it inside. So the commercially available media which is coming will have a expiration date of around 6 months to 12 months. But once you have added your serum and your other components, your antibiotic, now it is only valid for 1 to 2 months. So more than 2 months, do not store your media and use it later. If you have stopped your cell culture and you have a bottle which is already prepared using everything, and after 3 4 months if you want to start again, use a fresh media, make a fresh media, do not use the same old media, because your antibiotics or your FBS or the other things that you have added might have been degraded in that. The 4 degree delays the degradation, but it is not forever. Within 2 months you have to finish your media once prepared. Then avoid water baths from becoming dirty. Okay, so you should use some cleaning products. I'm not advertising this. It's just that you have to use some kind of cleaning products, disinfectants, because ethanol is very expensive and you get it with license and limited amount. Maybe for these kind of surface cleaning, you can buy surface disinfectants that are given by companies like Sigma or Thermo or some other companies. And keep everything in the cell culture very 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 clean. So your water bath where you thaw your cells, when your frozen vial, you take it, you thaw it in your water bath for 37, or you have to pre-warm your media before using for 37 degrees. So these water baths are the source of contamination. Wherever there is moisture, there is source of contamination. So these days people are using these metal balls, heat bath. So they, instead of water, they have metal balls. The heat generated uh, ho- will be transferred to the metal balls, and the metal balls will be holding the heat. And then you place your bottles inside those metal balls, like that. They're like marbles, they look like marbles. So that is much better, it will completely avoid contamination. If you're using water bath, then immediately frequently keep changing the water, keep it clean, keep cleaning it. Okay. And anything that you have put in the water bath, immediately after taking it out of the water bath, spray it nicely with ethanol if possible. Dip it in ethanol if they are small tubes like your vials, etc. Big bottles you can spray it with ethanol and immediately clean them. Last, don't allow essential equipment to become out of calibration. Ensure microbiological safety cabinets are tested regularly. So, your instruments or your cabinets, your airflow chambers, your microscope, if you have a digital microscope, your incubators, all these things should be calibrated at their given amount of time. If not, it might seem that they are functioning properly, but in reality they won't be functioning, and then you might be thinking from where the contamination is coming or why my cells are not growing. So your incubators should be regularly calibrated, your airflow chamber should be regularly calibrated, the air pressure should be checked, your valves through which your CO2 is pumped should be regularly checked. So all these things are very important. Let it be reagents, let it be instruments, everything should be properly checked and cleaned at regular intervals. So these are your do's and don'ts for your initial cell culture start. And if you have any doubts in these things or questions in these things, you can ask me. Anybody? I hope it was useful and helpful. Okay, if you guys don't have any questions, we can end the session here. Thank you for attending.